Today I will be explaining the different type of point mutations that can occur in a gene sequence. A point mutation is a substitution where a single nucleotide base is changed, inserted, or deleted from a sequence of DNA or RNA. This mutation can be described as either a transition, which is a substitution for a purine with a purine or a pyrimidine with a pyrimidine, or it can be described as a transversion, which is a substitution for a pyrimidine with a purine, and vice versa. If this mutation occurs in the coding region of the mRNA, then the amino acid sequence could be altered, which affects the proteins produced. First, we are given the coding strand of a DNA sequence, which was transcribed into an mRNA sequence and then translated into an amino acid sequence. This represents the normal and desired sequences, and for this presentation, let's say that the normal amino acid sequence here is the word adapter, so keep that in mind during the next examples. So we still have the same DNA sequence, but now there is a mutant mRNA sequence that represents a missense mutation. A missense mutation is defined by a base substitution that changes the amino acid, which could lead to changes in the protein structure or function. This is also a transversion point mutation because the base is changed from a purine adenine to the pyrimidine uracil. Now after the missense mutation, the word adapter has changed to adapted, which has a different meaning. Next is a mRNA mutant sequence with a nonsense point mutation, which occurs when a base substitution leads to an early stop codon in the amino acid sequence, resulting in a shortened protein. This is a transition point mutation because the pyrimidine C in UGC was changed to the purine A, UGA. Now our word adapter has a period in the middle of it, representing an early ending before the end of the word. This slide has the silent point mutation example, which is a base substitution that does not change the amino acid reproduced. This is a transversion point mutation because it is pyrimidine to purine. Now our word adapter becomes adapter, which is spelled differently but translates to the same definition. For the last point mutation example, the amino acid seems to not have a stop codon. This is called a read-through point mutation, where the base substitution changes the mRNA sequence for the stop codon, resulting in a longer protein. This also shows a transition mutation from purine to purine. This might be a stretch, but now our word adapter could become adapted education, which shows the last letter or the stop codon of the word being changed and then continued on. I hope these words help to better understand point mutations in gene sequences. Thank you so much.